Hey guys, welcome to topic 3 in chapter 20. We've already talked about how we build tissues from cells. Now we're going to talk about how we maintain them and how we renew them. Here's our topic outline. This is going to be a pretty short lecture, so hopefully we don't go too fast, but if you need to back up or if you still have questions, just let me know when we get to class and we'll talk about these some more. As always, here's our topic objectives. These are what I expect you to have mastered before our next exam. Alright, so let's go ahead and get started. So we talked a lot about what tissues are, we've talked about how they form, we've talked about the junctions between the cells. Now let's look at them in a little bit broader of a view. And in this case, we're going to start out with tissues and cell types. So it's easy to think that a tissue is formed of all one cell type until you start really digging in and focusing on it. And you realize that no, there's actually a variety of cell types within there. Not only are there different types of cells, but there's even different types of tissues within the same tissue. For instance, you can see that we have a variety of connective tissues in the skin, as well as blood vessels and the epidermis, and this whole thing just comes together to make the tissue and give it the specialized functions that it needs to do the process it's been built for. And all of this and how the tissues are developed has come through the processes that we've talked about all semester. So everything we've talked about prior to this is what comes together to form these tissues and cell types and give it the unique function that it has. And the entire process of development and because we all start out the same, all animals, all organisms at one point is a single cell, and so we don't quite understand exactly still how all these different organisms come out of a similar original structure. We know it has a lot to do with genetics, and we know it has a lot to do with regulation, but how and the processes that have to do with this are not completely understood. We do have some understanding, but not a complete one. So it's important to understand that this is still a really big growing area, and developmental biology is a huge field right now. So tissue has to be renewed. And we're going to talk about that here in a second as to how that happens. But as these tissues as these cells die through apoptosis and other things, we have to replace them. And how do we maintain the cellular organization and the function of these tissues? Well, there's three main things that are used to help maintain the cellular organization of these tissues. The first is cell communication. We've talked about this a lot, where signaling between cells will help maintain the function and the events that are occurring within the cell. Then we also have a selective cell-to-cell adhesion, and this helps keep the cells together because the cells like to bind to similar junctions and similar structures. That helps ensure that new cells will bind to their neighbors and continue to form the tissue that we've been talking about. And the last is cell memory. It would obviously be a problem if we had a fibroblast that differentiated into a neuron, and that would be a problem. So the cell memory is really important, and that the fibroblast is going to divide into a new fibroblast when it's needed. And so that cell memory is maintained so that the tissue maintains a history and the same cell components it did when it was first developed. And so all of this leads to that stability of the tissue that we've been talking about. So tissue renewal, as we know from our skin, it flakes off regularly, and this is because a lot of tissues have to be renewed. And some tissues don't renew it in the same rates as others. As we know, some cells are in G0. Some tissues have to renew a lot faster, especially such as those in the gut. Those have to be renewed about every 10 days or so. And there's significant variation across the entire body. Some tissues renew really quickly, some renew really slowly. And this is a very important process. If we were to knock out this process and prevent tissue renewal, it could result in the death of an organism. So it's really, really important that you understand that renewal is a key factor to all of our tissues. And cancer is actually, we're going to talk about this in topic 4, but it arises from the lack of renewal controls, where cancer cells will just keep going. They don't actually stop. So renewal is controlled through not only the division of cells, but also the death of cells. And if we don't have these renewal controls, the cancer cells can arise and form a tumor. So this process of renewal all starts with stem cells, and we have these dividing stem cells that create precursor cells, and these precursor cells then divide into the different types of cells that we need, and they all move through the process, through the structures, and renew the tissues as they are needed. And you can see that here with the gut cell example. And as I said, the replacement process varies depending on tissue type, and it's controlled very tightly through different molecular signals. And this is both partially from internal molecular signals and external molecular signals for the environment. One of the key proteins and key pathways in cell renewal is the Wnt pathway, WNT. These proteins help stimulate it and can be dispersed to other cells within the area that helps signal that we need to regenerate or not, depending on which part of the pathway we're talking about. But this is an important pathway for this. So let's wrap this up by talking about therapeutic cloning. So we've talked a lot about this in class throughout the semesters. How do we treat disease and how do we handle these kind of things? And so therapeutic cloning is one area that we're looking at for this. Now let's go back to our basic definition of cloning. And cloning is cells or individuals that are genetically identical. And so in this case, we're going to talk a lot more about cells. We talked about reproductive cloning once upon a time. That's where we're going to generate a whole organism. But therapeutic cloning is where we can generate personalized embryonic stem cells from, or the idea of it, from a unfertilized egg and the injection of DNA into it, which would allow us to create essentially new embryonic stem cells. The importance about embryonic stem cells is that they're pluripotent stem cells, which means that they can go and develop into anything. So if somebody needs a new heart and we could develop this process, we could grow them a new heart on some scaffolding with cells that have their own genetic markers, so we wouldn't have to worry about rejection. However, this has not been successful in humans yet, so this is all just hypothetical. We have successfully done it in a few other species, but not in humans. One thing that is relatively new that has been developed is the idea of these induced pluripotent stem cells. So what they have found is that they can take a cell from skin, for example, and they can introduce 3 specific genes that will essentially reverse these cells and take them back to a stem cell type status. And this is known as iPS. And it hasn't progressed a whole lot right now because it's pretty new, but that is how but this is a new cutting-edge frontier in how we can help repair damaged tissues that need to be replaced. So this is the end of this topic. As always, if you have any questions, please let me know.